Okay, so human papillomavirus multiplication cycle, or this goes for all papillomaviruses. So these viruses are tumor viruses, just like the Papillomaviridae. So you're going to see a lot of similarities between the two. This is just like the case for Rhabdoviridae and Paramyxoviridae. They were very similar. Same thing here. Papillomaviridae and Papillomaviridae are all very similar, and you'll see why. Okay, so here again you have HPV as a double-stranded DNA virus, which means that it will need to wrap its DNA around histones, and these histones are the exact same histones that we would have in our cells: H2A, H2B, H3, H4. Now it's a bit bigger than Papillomaviridae, which is usually around 5,000. HPV is 8,000. So, and then you have the capsid proteins here, L1, which is major, L2, which is minor. From my understanding, the L's and the E's, like right here, that you see, L stands for late, and E stands for early. So this is like a late 1 protein that is the major capsid protein. Okay, so you also have the exact same adsorption, which is, my eyes read it as it attaches to the receptor. And so it attaches to the receptor, so just like any other multiplication. Well, let's see, this is alpha 6 beta 4 integrin. Let's go to Google Chrome and see what's like for. So SV40 is MHC class 1. Okay, so there's one difference between the two. Sorry for all the switching here. So there's one difference. So here's one difference between the two tumor viruses. The cell receptor that attaches to. The other one is, of course, the capsid proteins. But anyways, other than that, it's still very much the same so far. So attach, penetrate. You have that partial uncoating like before. You have to transport into the nucleus. And I'll reiterate, it has to go into the nucleus because it's a DNA virus. So when it makes its mRNA or replicates its genome, it has to do so in the nucleus, where the cell machinery where the cell machinery can actually read DNA. You have encoding. So then, you have full encoding right here. So look again right here. That looks oddly similar to this one for SV40, for the papillomavirus. So again, I'm just trying to illustrate the fact that they're very similar. They have an early, they have genes that are transcribed early and genes that are transcribed late. Now the only difference is that, or the difference between the two genomes is that for Papillomaviridae you have bidirectional regulated transcription, right? Here it's all unidirectional. So it doesn't transcribe one side counterclockwise first and the other side clockwise. It just all goes clockwise. So it'll transcribe these early genes. Excuse me. And basically, when it transcribes the early genes, it makes, it transcribes it using a cellular RNA polymerase 2, and also using its own E2 protein and the GTFs, which I forget what GTF is, or if I was to do it off intuition though, it would probably be something related to cell signaling. And it probably uses guanosine as energy or something. Long story short, you transcribe the mRNA and then you splice it like normal because it's DNA. The mRNA transcript is a transcribed DNA. And if you want to make the proteins, you gotta make it go outside of the nucleus, because that's where the ribosomes are. So here in the cytoplasm, some of the genes undergo splicing right here. Others are just translated completely, like normal, no splicing. Now the important proteins that I want to focus on for like the next 30 seconds or something is E6 and E7. E6 binds to P53, E7 binds to RB. Essentially, what binding to those two things do is they tell the cell to not kill itself. It prevents apoptosis, and it drives the cell it tells the cell to start growing. And with growth, you get DNA replication and protein synthesis. And these two proteins, E6 and E7, they have to go back into nucleus where it can finally tell a cell to start growing. So now the conditions, so this E6 and E7, I think I see them as the analogs, the papilloma analogs of the polyoma T antigens. They go back into nucleus, tell a cell to not commit suicide, tell a cell to start growing. So now it's back in here. Conditions are now the infrastructure to replicate DNA and to make huge amounts of viral protein is there. So here you have a bunch of progeny DNA. From that, again, you have temporal or time-based control of late transcription. So you transcribe only after you make progeny DNA, which makes sense. Because the late proteins code, L1, L2 are the capsid proteins. And you don't want to make capsid proteins until you have viral proteins for the capsids to encapsulate. Now late transcription, splicing again, transport the splices out into the cell or into the cytoplasm. You have translation of your proteins, blah blah blah. These go back into the nucleus, right? Because you have progeny DNA, progeny viral DNA, which cannot leave the nucleus because the nuclear pores are too small. So you have progeny DNA plus capsid proteins that were imported into the nucleus. And look, now you have your assembly. It gets exported out into cytoplasm. It's enveloped for a while, and that envelope comes from the nucleus, and you have viral release. So very, very similar, except for maximum 5 things, if not less, that are different from polyomavirus. Honestly, if you get polyomavirus, you'll understand papillomavirus. So hope that helps. See you in the next one.